let's get started. Okay, so Megan and I are going to be talking about two new pilot programs from MASERC and the AIS Detectors program, focusing on spiny water flea surveillance and zebra mussel safaris. They're called zebra mussel safari and spiny surveillance. So first of all, want to introduce ourselves. It is totally understandable that you might think that we're the same person, because having two Megs, both working on AIS outreach and education and affiliated with MASERC, is confusing, especially since you probably primarily interact with us over email or Zoom. But fun fact, we are two different people. I'm part of the MASERC administrative team. I've been here since 2020, and I mostly work on research translation and implementation. Megan works for University of Minnesota Extension as an AIS educator, and she's been doing this since 2016. Megan's work is primarily focused on developing and managing AIS-focused extension, citizen science, and public education programs. That said, there's still a ton of overlap in our work and the people we work with, and we team up on a regular basis for various projects and initiatives, including what we're going to talk about today. So unless you're writing our paychecks or grant reports, the distinctions aren't all that important. We just thought you wanted to know you got a Meg and a Megan. And now let's introduce our target species. If you are attending a MASERC showcase, you're probably very familiar with these already. Spiny water fleas and zebra mussels are some of our most challenging invasive invertebrates in Minnesota. They have high impacts, they spread easily, and unfortunately our control options are limited to non-existent. And we have some significant knowledge gaps for both species that impede our progress in prevention and control. And these two programs you're going to hear about are meant to help us address some of those challenges. So I'm going to talk about spiny surveillance monitoring first, and Megan's going to cover the zebra mussel work. So what am I talking about when I say surveillance monitoring? In this case, our definition would be a system of surveys that's being implemented in an at least semi-coordinated fashion and implemented using strategies to maximize our detection of the target species for the purposes of increasing our knowledge about the distribution of spiny water fleas and detecting new invasions early. So this is a pilot project initiated by MASERC, but really the reason for its existence is almost entirely based on three previous MASERC research projects that I really want to briefly acknowledge here. So we have Valerie Brady's work on the highest risk activities that spread spiny water fleas between lakes. Don Brandstrader's work showing us that spiny water fleas may be present in lakes for many years before they're detectable. And then Gretchen Hansen's work showing us that young walleye and perch in spiny water flea infested lakes are significantly smaller than those from uninfested lakes. So this program also builds upon the energy and connections local managers and lake associations that we forged during last year's Stop Spiny outreach campaign. And one of the biggest takeaways from working with these folks all over in Minnesota was that we really needed better information about where the spinies actually occur. And if we have that, we can better target a potential future campaign to the highest risk parts of the state, and get that more targeted for local managers to figure out which lakes in their county actually have spiny water fleas. So this is a program that's going to be designed to equip volunteers and local managers with the knowledge, the gear, and the right reporting methods to help us optimize the detection of spiny water fleas. And so this idea was really hatched out last fall between me and Don and Valerie. And with their work, primarily, we really had the why, the how, and the when to inform development of the program. And of course, they both have extensive experience with study design and doing zooplankton surveys in the field. And that really helped us tailor our program to be suitable for volunteers. I also want to acknowledge Gary Montz, Kylie Couture, and Jake Walsh with the Minnesota DNR. They have been extremely supportive of this project and provided a lot of input through all stages of its development, and especially when we finalized our monitoring handbook. So shout out to the Minnesota DNR for their support. So let's get into the why. And I want to note, I'm mostly going to be talking about why we should be doing surveillance for spiny water fleas and the resources that we've put together to support this, rather than the actual surveillance methods. That's kind of a whole other presentation that I can easily share with you if you're interested. So number one, the fact that we only have spiny water fleas so far detected in a pretty small number of water bodies, 67 so far, and only one new lake has been added to that list since 2016. And if you think about how many lakes are added to the infested waters list for zebra mussels, Eurasian water milfoil, starry stonewort, it really kind of points to perhaps a detectability and recognition problem rather than spiny water fleas not actually spreading. So, and one of the reasons why we're maybe not knowing about new spiny infestations is because there is really no formal monitoring happening in Minnesota for spiny water fleas. And I should point out that spiny water fleas are not just a northern Minnesota problem. Temperature studies, temperature modeling, and knowing spiny water fleas temperature thresholds tells us that spiny water fleas could survive in many of our lakes in Minnesota. And one of the most severe spiny infestations in the upper Midwest is actually in central Wisconsin, in Lake Mendota near Madison. So that tells us that it's not just a concern for northern Minnesota. The other reason is that we know that spiny water fleas spread easily through the recreational angler and boater pathway. This was the study that Valerie Brady led, testing the highest risk vectors. We found that spiny water fleas typically get caught on fishing gear like monofilament line, other tackle, like the pulleys we see here, fishing pole eyelets, and they are small and difficult to see and can easily evade boat inspection. And this shot on the right is from our AIS Explorer tool, and it should highlight the concern here. So Mille Lacs is one of our sort of outlier infestations, and Mille Lacs, of course, is a tremendously popular fishing lake. And this network that we're seeing, all those lines leaving the lake, is the outgoing boater network. So if you think about all the people leaving Mille Lacs with their boat and fishing gear and where they're going next and the potential spread of spiny water flea, it's quite concerning. So another reason to do this is spiny water fleas are a really high impact AIS. They're predatory zooplankton. They have few predators in our lakes. They're basically inedible to a lot of young fish because of that tail spine. And they consume massive amounts of native zooplankton with effects to food webs and water quality and clarity. They have early maturation and explosive growth and a very adaptable, flexible reproductive strategy. And there are no control methods currently available or really even in the research phase. So this all just highlights the importance of prevention. To be effective in prevention, we need to know where they are. So hopefully you're convinced by now that increasing our knowledge of spiny distribution is worthwhile. One of the other reasons that we should do surveillance is because we can. These are not like zebra mussels where you need to send a water sample to get a Veliger test done, and it has to be done by a specialized scientist with a microscope. Spiny water fleas are small, but they're not microscopic. You can see them with the naked eye, and it helps even more if you have some really basic equipment like a white plastic tray, a fork or a forceps, and a magnifying glass and a headlamp. This is all stuff that's very approachable and attainable, and with a little bit of training, you can go out and survey for spiny water flea yourself. So these are some of the resources that we have set up for the program. We have a whole web page set up for this. Don Brandstrader recorded a series of short videos showing you how to deploy a plankton net, how to sort your sample, how to document a find. That's a really key thing here. We, of course, the first people you contact is the Minnesota DNR, and you're going to want to save a sample. And then we also have a records tracking system. Don also made a video about native lookalikes to help you effectively identify spiny water flea if they're present. So that's available on the site. And there's a user-friendly manual that has all the instructions you would need, as well as a gear list and really important guidance on how to time your surveys, the best time of day and the time of year to increase your chances of detecting spinies if they are present. And this is all based on work that Don Brandstrader did on seasonal abundance patterns of spiny water flea. And we also ran some customized models with the AIS Explorer modeling software that we have to help to provide county managers with lists of lakes that would be prioritized to do surveillance in their county. So we're trying to help you figure out the highest impact places to search and when and how to do those surveys. And I included this little cutout of the spiny water flea anatomy to indicate, just to remind us that they're small, but they are totally visible. And that black eye spot is really distinctive among other Minnesota zooplankton and will help you identify them if you have the right equipment. And then reporting is really important. As I said, first people you talk to and notify is the Minnesota DNR, but we have also set up a web application. Alex Badges helped with that. He's become our Shiny app guru in-house. And this is designed to help record all the survey effort happening because we want to know who's out there surveying lakes, and keeping track of the survey effort will help us better hone a surveillance program in the future to know how many surveys does it take to detect spinies at low density. So this app is pretty user-friendly, and it's available for anybody who's doing that work. And the last reason why you should do this is because it's fun. It's really cool to get out there. You might get out on the lake at a different time of year, different time of day, and it might be a whole new experience for you. I know for me and many others that I've worked with, when you do a zooplankton sample and you look at all these amazing little creatures under a magnifying glass, it's really eye-opening, and you might see your lake in a whole new way. Those two images in the middle, that's a glassworm at the top, and then the Leptodora. Below that are some of our more common native zooplankton that you might see, and they're really amazing creatures. So we think it'll be eye-opening. If you're interested in learning more, go to that website, Spiny Surveil. There's just a Google form to sign up, and I'll follow up with you with some links. I can send you a recorded webinar of the how-to and all the other resources. And we already have a number of local government managers in Cass County, Cook, Lake, and Lake Association folks out there monitoring already. So I'm getting good feedback from folks, and clearly from these pictures, they are enjoying themselves. With that, I will turn it over to Megan.
All right, great. Yep. So I'm going to take a chance to talk about the other project that's in development. As you'll learn as I go through this, it's at a very different stage than the spiny water flea project that Meg just got done talking about. But again, the team here, Meg and I again, have been working on this along with Pat Mulcahy, who's another member of the extension program team. And then Keith from the Department of Forest Resources has been brought in the kind of the phase 2 of the piloting, I suppose, to help us with some hopefully much better ways to process our data. And I'll talk about all of that as we go. Oh, do you want to do the slide? There we go. Okay, so what is this project? It's still in its pilot phases, but our goal here is to create this participatory science project that gives a way for lake groups, lakeshore homeowners who are kind of stuck in this space with a zebra mussel infestation there, but kind of not much to do about it at this point in particular. This is a really great way for them to help contribute data that will maybe someday lead to some answers. And that data can then become available to researchers, resource managers, agencies, perhaps that lake group has questions about what's been going on in their own, the infestation in their own neighboring waters as well. So we're hoping to kind of have some data that can help answer these questions. So the why to this is, so we have a number of infested water bodies in Minnesota with zebra mussels. So you can see all the little yellow dots on the image here from the USGS curated map of infested waters. But some of the things that we lack, not only in Minnesota, but really kind of throughout the invaded range, is that there's not lots of long-term data, in particular data that's kind of readily accessible. And there really aren't many like large data sets that have data that's been consistently collected in the same way, so the same type of data across many lakes and over many years. This is going to be like a bigger data set. And the reason for this is like it's a lot of work, and it's hard for any one research team to be able to collect these really big data sets. And the way to kind of help with that is to have programs like this where lakeshore homeowners themselves can help kind of contribute this data in. And it can help make these data sets that we hope will be very useful to future projects. You can go to the next one. Okay, so this is how it works. And I have this really giant asterisk next to that because, like I said, this is a pilot. And we've been working through a lot. We've gone through 2 pilot seasons of this now. And we're making great progress every year, but this is how it works, as of today. And we're learning a lot. We haven't even finished the pulling plates from this year. I fully anticipate changes before we do this again. But what we've been doing so far is working with a small group of pilot lakes in Minnesota that have really active lake associations. So we know that there's like a good group of people that are organized and can, hopefully work together to do the set of protocols that we've created. And these lakes also have varying degrees of infestation severity. So some are like pretty new to have been infested within just the last couple of years, and some of them have well-established populations. And we're working with them to hang these samplers. You can see that with the 3 gray plates hanging out. We're working with them where the homeowners are then hanging these samplers off of the docks. And we work to kind of make sure that they're spread somewhat equally around a lake. So you can see the example from, I don't know, which one is this, Bay Lake? This is Bay Lake. You can see that we've kind of made these zones and said we really like to have at least 1 sampler within each of these zones so that we're kind of getting a good overall picture of the lake. And then we're having volunteers photograph the settlement on those plates at the end of a season and submitting that to us. And that's where a lot of the changes come. Some of the things that we've been working with are, like, do we really need 3 plates? Lots of time you see these samplers with 4 plates. Is 3 even necessary? Maybe 2 would get us the same kind of data that we need, and we can save on effort and materials and such. So that's one of the things that we're working on, but that I think is kind of minor in comparison to some of the other stuff that we've been working on. So I think on the next slide there should be here we go. So kind of the process that this then works through is, we have these many volunteer data collectors across the state, and many, it's still many right now, but again, this is a pilot. So we have about 5 lakes that we've been working on this year. Those volunteers are collecting this photographic data and they're uploading that into the MACERC cloud. And I'll talk more about that too. You've seen kind of like a preview. Also, it's very similar to what Meg just spoke about in the Spiny Water Flea project. This is kind of the new piece that was added this year, and I'll talk about that more too, is we have, hopefully there's this computer program that's going to help us spit out some numbers really quickly. And then this leads into this beautiful, large, long-term data set. But this is all, like I said, a pilot. So next slide. What do we mean by that? One of the things that we've learned is that it is perhaps similar to being a social media influencer. It's harder than it looks. You need the camera, you need an internet connection, and you need a lot of time on your hands. So we've been learning all of this, and the various things that you need are perhaps needed by other people. But some of the challenges that we've been kind of learning about this year relate to, the camera maybe is the easiest thing here. But there's an internet connection issue. Not all of the lakes that we're working on have really great cell service. So you might need to figure out a way to like upload things over Wi-Fi, and even then, not all of Minnesota has the best of, like high-speed internet. And then on the counting side and the processing side, we're noticing some spots that take a lot of time. And a lot of that relates to kind of this new development that we've been working on with how we're processing the plate. So if you want to go to the next slide, we'll kind of get to kind of how we've gotten to where we are now. So in pilot season 1, so this would have been not this summer, but the summer before, this is what the documentation looked like. So volunteers took these photos. They could mark what surface this was on that stack of plates that we had, and then they sent these photos into us. And then the goal was, well, this is great. All the data gets collected, and we can just kind of count from an image and get data that way. And then we looked at some of these photos, and like, this looks really difficult to count for a number of reasons. If you want to go to the next slide. The first thing that we learned was if we let the plates dry, it was much easier to see what was going on that plate than if the photos were taken wet. So the same plate, wet and dry. The other issue that you can see here is there are a lot of tiny brown specks on this plate. And pretty much all of them are zebra mussels. But you may not be able to tell that from here. And even zooming in for some of those tiny ones, if you just may not have enough pixels to really see the shape and to get a good clue on what it is. There's some challenges here, not to mention how long this takes. Like, I counted this plate by hand on site, and this one was a challenge, and this isn't even the worst of them. Okay, so then next slide kind of goes to what we started to do next. So then as we identified kind of this challenge of like, how are we still going to process this data, we got connected to Keith, who's done some really cool work on using computers to identify like invasive species and other bits. And he's largely worked in forest resources, but he said, yeah, I think a computer can do that. And I am not going to pretend to really understand how the computer does this and knows this, but I'm going to talk just a little bit about kind of the things that he's shown us and how he's explained it to us. But you can see here, there's an original photo. So if we were to just try and count manually off of that, you can see some challenges. There aren't quite nearly so many like really itty bitties here, but there's some stacking and some clustering that can make it hard to see. But as he put this image, this is one image from the top down, through the computer, he can apply various filters on there. So the one shows kind of the color banding, so reds and blues and greens. And he's like, oh, look, like the zebra mussels tend to have more green than other things. Okay, so this is one thing that the computer can do to identify and count. And then also zebra mussels have a pretty distinct shape. So he runs this filter on it that relates to edge detection. You can see kind of the red speckles on this plate, and this is kind of what the computer is seeing, for lack of a better word. And it can then use that information. So as it tries to layer things on, the hope is that then the computer can just look at this image and count the mussels for us. But I had mentioned the stacking. So there's some challenges that we found here. Like this is looking like something, but we think maybe we can do better. So on the next slide, we learned that like maybe 3D imagery can help with some of that. So this would be like another layer that gets stacked on. So now the templates look a little bit different, where they have these colorful corners. And the reason that time and internet have become a thing really this year is that to get this 3D image, we learned it takes about 20 photos of a surface. So the colorful corners help the computer align all of the images together, and then it can stitch all of these images together and create something that looks kind of like what you see in the gray image. And that can kind of help identify stacking. So it's another piece of information that the computer can use as we're trying to tell it to count things for us, that we don't have to spend all the time doing it. Because I don't think we could just feasibly do many plates in a year. Okay, next slide. Okay, so that's pilot season 2. You can see a better image of like the true template that was being used, and you can see volunteers taking like all these different angled pictures. They're getting that original top-down one, but then kind of going up and over and around, trying to get all the possible angles. And then Alex has also come to the rescue as well, and created an R Shiny app for Zebra Mussel Safari so that people can upload those images and it'll automatically tag the images for us with the data that we need to then send that to Keith for the computer program that he's working on to do this work for us. And that's the piece that kind of takes some time, is if you have 3 plates and you need 20 images per surface, there's 2 surfaces per plate. So all of a sudden that's 40 images per plate, and we're at 120 images for 1 stack of plates. And then if you saw on Bay Lake, there were 20 something plates, I think, on that lake, or maybe it was in the teens. I forget where it landed there, but it's a lot of images. So in particular, if you don't have the best bandwidth, this can take some time. So these are the things that we're kind of working through now as we move into this next phase. And then next slide. Okay, so that's where we are. So what's next? So I've mentioned a lot about challenges, and I think, and that's where we are. So I have a pair of 6-year-olds. So like if the song is now flowing through your head, the kind of just keep swimming is where we are. And you know what, we're trying to learn a lot and figure out like, how can we make the process easier? What are we going to learn from like how these computer counts actually work? Like maybe ultimately we don't actually need the 20 photos, and things are looking okay. And we can cut things like that down, or maybe 10 would still give us the same amount of data. Maybe there's other ways that we can do the photos. Maybe there's other ways that we can train to have these happen. Hopefully we don't need to hand count nearly as many as we think we do to kind of ground truth the computer to it, because it turns out that's taking a really long time too, because many of these mussels, this is ultimately all measuring annual recruitment. So we don't see many of the really big mussels. It's lots of tiny mussels. And we're talking like grains of sand, tiny sometimes. So it's really difficult to kind of process those plates. And we're hoping that the computer program can ultimately handle that, and we can find a feasible way for volunteers to be able to collect that data and upload it over for the computer to make everything easier on all of us. And that's, oh, I think there's one more slide. There we go. So this was also our thank you to the lake groups who have helped us and who have been a part of the pilot. In pilot year 1, we just had 3. So Sharon Natso with Long Lake Association, and Karna Lundquist at the Big Lake Community Lake Association, and Kevin Zahler at Lake Minnewaska Preservation Association have kind of been our point of contacts. And there's many of people who are volunteering time and dock space and all of that on those individual lakes. But these are the ones who are kind of, I think, taking on the bulk of the workload as it relates to making sure this is able to happen on their lakes. And then this year we added in Lake Latoka, so Chad at the Lake Latoka Association, and Phil Rollins at the Bay Lake Improvement Association have been new this year with kind of the new bits. So we tried to add more lakes to make sure we're able to get kind of the full variety of things that the computer might encounter in the future in this pilot. So thanks to all of them for all of their help and work in as we kind of keep working on the development of this project. And that's all that I have. So we have, there's Q&A next, and my box just disappeared.
Okay, we got a question from Naomi, and I'm going to defer this one to you. Is there any reason for using dark gray plates versus white plates? It would seem that white plates would make it easier to see the mussels for both people and computers.
Oh, yeah, that's a really good question. So we use gray plates because if given the opportunity, mussels prefer darker surfaces over lighter surfaces. So it could be interesting to test the two and see if we can still get enough settlement on the white to get our data to. I don't know, we can throw in more things to try, and who knows, perhaps another pilot season of it. But yeah, that's the reason that we collected the gray plates, or that we started out with gray plates.
Any other questions? We have a little bit of time. Oh, all right. What is the texture of the plate? So these are just PVC plates, so it's fairly smooth. Megan, anything you want to add about the plate texture?
Yeah, I think that's it. It's a smooth PVC. Like, I mean, if you just wanted to do like early detection with them and it wasn't necessarily part of a project like this one where we're looking for like a consistent sampling device and method, it doesn't matter if it's textured or smooth. Both would be fine. I've used like a textured, like an ABS, a different type of plastic before too, and they settle on that just fine.
Yeah. A couple of sort of questions here on timing and what's happening next year for both projects. I'll try to address those. So spiny water flea surveillance. If you're someone who signed up and maybe you haven't gotten your net yet, or it just didn't work this year, that's okay. I will note, however, that it's been a really warm fall. And even in quote unquote normal years, we might see spiny water fleas into October. So there is still time. And this is still a really good time to survey for spiny water flea. If you are super jazzed about spiny surveillance, send me an email right away. We worked with a company called Aquatic Research Instruments, really great little small business that makes high quality sampling gear. It's kind of a one guy shop in Idaho. He made several dozen of sort of these custom nets specifically designed for really efficient spiny water flea sampling. They're called student sampling nets, but they're designed for spiny water flea. And he made like a dozen extra. So he has some ready to go, ready to ship. And I can give you the information for that. So there's still time this year. Spiny surveillance, I see no reason why it wouldn't continue next year. We'll, all those resources will still be available, and we will offer more webinars to get people trained up. As for zebra mussel safari next year, that's quite a bit of a question mark. We really need to hear back from our imaging guy, Keith, to kind of see how things looked with this new method that we're trying this year. And really need to kind of assess some big picture feasibility questions. But I will, I'm going to put my email, or Megan, would you mind putting both of our emails in the chat? If you are interested in spiny surveillance, you can email me. Or go to the website, the Spiny Surveil. There's actually a Google form where you can sign up, and then I'll have your information there. I will follow up with you. We don't have a form like that set up for zebra mussel safari, but just reach out to both of us, and we will get you keyed in if we do scale up next year. It's a big if right now.
Yeah, maybe so what we've looked for so far for like a pilot group has been lakes that have zebra mussels. And again, I mentioned that it has like the varying levels of severity, or maybe like varying time since first detection. And we've learned, like, especially for this pilot, it's really key to have at least one person there who is willing to kind of take charge and be the point person for the rest of the lake association. People have done two different ways of collecting the data. So on some lakes, the person whose dock the sampler is deployed on is doing the photographing and setting things in. And on some lakes, that point person is maybe working with another person or group, and they're just going around and doing it all for everyone. And either one is fine with us. So whatever works for that lake group would be fine. But as we look at it and see if we're going to expand again, if like 5 is kind of where we need to be, we're certainly happy to at least have other lakes to consider as we kind of make those decisions about what the next step is.
Yeah. And just thank you, everybody, that a lot of offers in and interest to help. We really appreciate that. One of the questions about, are these programs designed to be self-guided, or do you need to attend an in-person training? In-person training is not required. When we did a couple online trainings for the spiny surveillance, that was just me and Don and Val giving a webinar. We had a short presentation, and then we did some Q&A. The instructional videos and the manual really should be enough. It's not rocket science. And I am here, to offer support and expertise if you run into snags. Dana asks about the price on the nets. With shipping, it comes with a net and a little carrying case to protect the mesh. I think it's about $320. So it's not nothing, but for a high quality, aquatic sampling device that if you take care of, you're going to be able to use for many, many years. I think it's worth it. And if you get some folks trained up, I know some lake associations send water samples to labs to have them tested for spiny water flea. It's not really necessary. If you have the training and some basic equipment, you can do your own surveillance for that. Oh, this is an interesting question from 9 Mile Creek Watershed District. Would it be possible to use older data without pictures? They have an Adopt a Doc zebra mussel detection program. Interesting.
That's a really good question.
Yeah.
I think the answer right now is I don't know.
Yeah.
I think it'll depend on how this data comes together, and then I think we'll learn more about what can feasibly be patched into it and still like make sense in the data set and how that ultimately reports out. So I don't think the answer is necessarily no right now. It might be, but it might be yes. That'd be really helpful. I don't think we know yet.
Yeah. Well, and someone is noting that they have black plastic samplers with a slightly rough surface, but difficult to see. All right. Well, you all have our contact information. You know that we're 2 different people, but if you screw up, it's okay. We will forward your information to the correct Meg. And really encourage you to check out the spiny surveillance resources. That's ready to go. And then stay posted about zebra mussel safari.